So properties of life. This is something that's probably, it's so rudimentary, I'm probably not going to have it on the final, but do you guys remember like the general ideas of what constitutes something as living? Can reproduce. You remember that? Can reproduce. What else? What else do we have? Can grow, uses energy. Remember that list from day one? Yeah, regulate. So it has the potential to regulate their internal environment. So it has the potential for homeostasis. Reproduction, can grow, use energy, can evolve. Now we've learned that, you know, individual organisms aren't going to necessarily evolve. The population of organisms will evolve. Organization. One general characteristic of living things is that they organize their internal environment in the presence of a chaotic external environment. Okay, we can move on from that. Be able to recognize a hierarchy of life in order from smallest to largest. So we're talking least inclusive to most inclusive. Let's just, and we're talking, let's see, recognize a hierarchy of life. I think for the purposes of the final, I'll probably start a little bit big picture. I'm not going to start quite at the cell. So we could just start at population, which is, do you remember what population is? All one species. And you guys could see the screen, right? Okay, yeah. Population, all one species, community. That's going to be multiple species. Multiple species. And then we have a, what comes after that?
An ecosystem.
Ecosystem. And that's going to be multiple species plus abiotic factors. And then outside of ecosystem, I think we just go straight to biosphere, right? Biosphere is Earth. All right, recognize the difference between deductive and inductive reasoning from definitions or examples. So just to put this in blanket terms, I would say that deductive, you go from large picture, so you go from bigger picture to smaller picture. And by that I mean you go from knowing the general laws that govern the universe and you apply it to something very small and specific. So I know that gravity is a law of the universe, and so if I take this manatee and let go of it, gravity will act upon that manatee and it'll fall. So you go from large picture wow, I can't spell, to small picture. And then inductive, this is where you go from small picture. So you're collecting data throughout your entire life. So let's say all of the pens you've ever seen have been pink. And so you use that small window of your own reality and you apply that to the universe. So you say all the pens I've ever seen in my life have been pink, therefore, then you make a generalization about the universe. Therefore, I conclude that all pens in the universe are pink. So you go from small picture, so our small window of experience, to large picture. Does everyone get that? Again, let me know if you need me to restart. Okay, so audio is staticky for Jane. Is the audio staticky for you guys?
Me too.
It's staticky for you too, Anna?
Yeah.
Okay. Turn down gain there, and then turn up gain here. How's that? Is that better?
It's better. There's still a little bit of feedback though.
There's feedback. Uh-oh. That's even worse.
Well, that might be the wrong word. I think static's still the right word.
Oh, still static? Shit. And now I'm getting audio clipping. Still bad? Two, yes. Oh, shoot. Yeah, I'm getting a lot of people. Frick. What do you mean? Oh, staticky. God damn it. This sucks. Worst time to have staticky audio. Let's try turning up the gain here and then turning down the gain here. How's that? Better? Worse? Still the same?
It's better on my end.
Okay, better. I'll work with better. We probably won't get optimal audio. All right. So that's deductive, inductive. If anyone has any questions about this, just yell that you're having trouble. All right, what are the parts of the scientific process? Be able to recognize parts from an example. Okay, this is probably something that we want to know moving on from this class, the scientific method. I should come up with an example on the fly. All right, so let's say one let me grab an example. Hold on a second. So what I want to do here is what I'm going to do is give you guys a series of sentences, and then what I want you to do is to be able to pick out what is the background research, what is the hypothesis, what is the conclusion, what are the results. So I'm gonna see if I can find something really quick. Okay. Oh, where did the thing go? Here it is. All right. What are the parts of the scientific process? Be able to recognize the parts from example. So let's just roll with this right here. So it's known that potatoes store nutrients in water. Okay. So that would probably be background research, right? What specific nutrients do they store? We remember the scientific method, right? So you have background research, you ask a question. So background research, then question. And then after your question, you try to form a hypothesis to answer that question. So 3 would be the hypothesis. Potatoes store their sugar as polysaccharides, such as starch. All right, so we have background research, then we have question, then we have hypothesis. Usually a hypothesis is a statement, not another question. If this is correct, then potatoes will stain purple with iodine. This is a prediction. So you go background research, then you go question, then you go hypothesis, then you go prediction. If this is correct, then potatoes will stain purple with iodine. Now 5, potato juice stain purple in the presence of iodine, like the known starch solution. So that would mean, or that would be the experimental results. So this is results. Water did not stain purple, but remained brown. So another piece of results. So 5 would be results, 6 would be results, and then 7, the data supports the hypothesis. So this is where you reject or accept your hypothesis. The data supports the hypothesis that potatoes store sugar as polysaccharides such as starch. So that would be your conclusion. So 1 would be background research, 2 would be your question, and then 3 would be your hypothesis, and then 4 would be potatoes will stain purple with iodine. That would be prediction. because you're predicting what's going to happen in an experiment. And then 5 and 6, those are both experimental results. Shoot, I ran out of colors here. Experimental results, and then number 7 would be your conclusion, because you accept or you reject your hypothesis. I'm out of colors. Or I'm out of colors, that makes sense. Okay. So if I were to give you an example like this, be able to pick out what's the conclusion. If I were to say, what's the conclusion? You'd probably say, well, let's look for a sentence that says the data supports or rejects the hypothesis. If I ask, what's the background research? Probably going to be the first bullet, which is, you know, usually starts with background research. Okay. Be able to recognize the control group and experimental group from an example. Okay. And this can kind of be grouped together with no difference between a positive and negative control. So a control group in an experiment is going to be the group that you make reference to. So let's actually just use the potato example. I have, I'll see if I can draw it out. Yeah, here we go. Okay, so I'm gonna pretend like this is a whiteboard. Annotate. All right, I'm winging this too. All right. Oh, we don't want a line. Okay. So let's just roll with the potato thing. All right. The potato thing. So we know that potatoes. What was? Let's go back to it and read it. Hold on. Potatoes store nutrients in water. All right. What specific nutrients do they store? We think potatoes store their sugar as polysaccharides such as starch. Okay. So this is our hypothesis. So one thing, and this is actually something that you would have experienced if we had a grounded class, is what we do is we take iodine. So iodine is actually very reactive with polysaccharides such as starch. What happens is when you throw in iodine and starch into the same solution, it actually turns this blackish, dark purple, blackish color. Now, naturally, iodine just on its own is like this lightish brown color. So when it reacts with something, it turns purple. We'll just say it turns purple. It's like a dark, almost black. So let's go back to the... So to run our experiment, we take 3 different test tubes. And in one of them, well, actually in all 3 of them, we're going to put, or actually no, in 2 of them, we're going to put potato. So we'll just say this is potato and this is potato. And then all 3 of them, they're going to have water in them, okay? Now, to determine, so when we're running an experiment and we know that iodine stains purple when it interacts with starch, what we want to do is we want to see what the solution looks like as a reference point. So the thing is, if we want to see if there's starch in potatoes and we want to use iodine as a teller for this, what we want to do is throw iodine in with something that is guaranteed to have starch in it, because that's our positive reference. That would be our positive control. And so what we do is we take literal starch, because we know starch interacts with iodine, right? So we take well, not literal starch, but we take starch. So we'll just make starch, you know, like a pink. So here's starch. We put starch into a vat. This is our positive control. And what we do is we add iodine to it. And we know that iodine is like this brown color. So we add iodine into the starch solution. and it turns purple. So we'll just put, you know, purple becomes the solution color. And so we know that that's a positive control. We know that this is what it's going to look like if our potatoes have starch. So starch, iodine, and I'll write iodine. Iodine plus starch equals purple. So this is positive control. All right. And actually, in this case, we're not putting. Okay. So we need to also know what happens, what it looks like when we add starch to no, or when we add iodine to no starch. Okay. So we're going to add iodine to no starch. We need to know what it looks like. And so we add iodine. Here's iodine to no starch. What happens? We get no result. We need to know what no result looks like because that's our reference point for no result. So this is going to be our negative control. Negative control. Now we have our experimental group, which is potato. We want to know if potatoes have starch in it. And so, well, what's our mode of recognizing if it has starch in it? We use iodine. So we add iodine to the potato. Now, if it turns brown, that would mean it has no starch. That would say negative results, right? Negative control. If it turns purple, then that means, well, it probably has starch. We know what iodine looks like when it interacts with starch, and so now we have a reference point. Now, when we happen to do that in the lab, we get a purple solution. So that would say, okay, so here's our experimental group. We compare that to our negative control and our positive control, and then we determine, you know, does it have a positive result or does it have a negative result? So this is our experimental. Wow. You get the point, right? Experimental group. Okay, so that's that concept. We have experimental group, which is the group that we're testing, and we compare it to our negative control and we compare it to our positive control. Most of the time when we're running biological tests, our negative control is just going to be water. Usually water doesn't react with much stuff, unless we're like doing some kind of experiment involving I don't know, the dissolving of solutions, then yeah, maybe water is probably going to play a role. But usually, you know, if we're running a biological experiment, water is going to be our control. It's going to be our sham group. You know, like you want to you know, give a nothing result. You're going to give them water. You're going to give them something that, you know, sugar pill or something. All right. Let's stop the drawing. Clear all drawings. Okay. So let's go back to the thing. All right. So that was these two. Recognize control group. Know the difference between positive and negative control. And also, like I said last class, all of these are our student outcomes. So the things that we're supposed to learn going into the class, like these are, I've plastered these student outcomes into our study guides. So I've crafted the class around the student outcomes, around the things we're supposed to learn moving on. Okay, so be able to define atom, element, cation, anion. That's easy, right? We remember that. We know this from our chemistry class we've taken. So atom, that's going to be basic unit of matter. It's not the most basic because you have subatomic particles, but we'll just say it's the basic unit of matter. Element, so an element is going to be a substance with only one type of atom. That's what an element is. And then a cation, so remember cations are going to be positively charged because cats are positive things. And then anion is going to be negatively charged because they're not cats. So an example of a cation would be Na+, right? Na+. So if you saw Na+ anywhere, you'd be like, that's a cation. If you saw Cl-, you'd be like, that's a negative charge. That's probably an anion. You could also associate the an with anti. Anti probably means negative, or you could associate that with being negative. All right. What charge are protons? And this is another student outcomes thing. What charge are protons, electrons, and neutrons? Protons, positive. Electrons, negative. Neutrons, neutral. Be able to tell how many of each when given a periodic table. So if I were to give you, let's just pull up a periodic table. Periodic table. I want one that shows us both the atomic number and the mass. Looks like we're only getting atomic number here. Oh, is that big enough? We can zoom in. All right, let's grab an easy one. So carbon. So we know that its atomic number is 6. What does that imply? The atomic number is the amount of protons. And then if you're looking at the atomic mass, what you do is you subtract the atomic mass, or you subtract the atomic number from the mass to determine how many neutrons it has. Now we know that if it's a positive or negative charge, it's going to have one less ion, or one less electron, or one more electron. So like, for example, you know, your C, where's your Cl? So chlorine, let's say you've got 17 protons. Now let's say it's Cl minus. That would mean that you have 18 electrons because you have one more negative thing than you have positive, making you more negative. And then with sodium, you know, if it was Na plus, that would mean that it has one less electron. So you'd have probably 10 electrons. What breaks them even is you have the same amount of negative things as you have positive things. So sodium in its neutral state, it's got 11 protons. If it's neutral, it should have 11 electrons. All right. Which elements are most abundant in life? Chan, right? So carbon, hydrogen, oxygen, nitrogen. Those are the most abundant. Now we have a lot of different trace elements that, you know, are abundant too, but those are the 4 most abundant. How are elements arranged in the periodic table? By their atomic number, or by their amount of protons, their number of protons. What makes atomic mass? Protons plus neutrons. What makes an element reactive? Their valence shell. Their valence electron shell. No difference between ionic bond, covalent bond, hydrogen bond. This is on the test. Ionic bond, that's going to be the stealing. That's going to involve the stealing of electrons. So sodium in its neutral state meets with chlorine in its neutral state, and chlorine has it's one short of filling its valence shell. And so what it does is it steals an electron from sodium, and sodium happens to have one extra. You could say one extra because it's closer to having nothing in its valence shell than it is to having its valence shell full. And so sodium gives up its electron, giving one more negative thing to chlorine, making it chloride, making it negatively charged. Now sodium has one less negative thing, making it positively charged. And then the disparity in their charges actually brings them together. So that's ionic. Covalent bond, that's when you actually have the sharing of electrons. So covalent is sharing, ionic is stealing. And then a hydrogen bond, this is where you have a partial positive on some molecule involving hydrogen, usually hydrogen, because they have such small nuclei. Because they have such small nuclei, they usually have less electronegativity. They have less of a pull towards the electrons. So when they form chemical bonds, those electrons have a propensity to move towards whatever other element they're bound to, or whatever atom they're bound to. So if 2 hydrogens are bound to an oxygen, those electrons are going to gravitate more towards that oxygen's nucleus, therefore giving them a partial positive charge, and that oxygen a partial negative charge. Now, the disparity in partial charges is actually what draws those hydrogens to oxygens on other water molecules. And so from that we get hydrogen bonds. So it's the disparity between two atoms with partial positive or partial negative charges. What makes a polar molecule polar? That's just simply having an uneven electron distribution and a lack of symmetry. So uneven electron distribution / lack of symmetry. Because we know that, well, I don't even spell symmetry. Because we know that if we have, you know, CH3 is the perfect, or CH4 is the perfect example, where you have that carbon in the center, and then you have a hydrogen, 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 hydrogen. It's perfectly symmetrical. You can't cut a line down the middle to where you have more electrons on one side or the other, therefore making it polar. Therefore making this non-polar, because you can't cut a line down the middle or make a line down the middle. Be able to recognize an acid or base from its pH number. So we know that low pH is acidic, high pH is basic, 7 is in the middle. So 7 is going to be neutral. All right, know the definition of organic. So what does it mean to be organic? That's going to be something that contains both carbon and hydrogen atoms. Know the monomers of carbohydrates, proteins, nucleic acid. The monomers of carbs are going to be sugars or monosaccharides. The monomers of proteins are going to be amino acids. And then the monomers of nucleic acids are going to be nucleotides. Know the difference between triglyceride and phospholipids. So a triglyceride is going to have a glycerol backbone with 3 fatty acid tails, whereas a phospholipid has 2 fatty acid tails, and instead of that 3rd fatty acid tail, it has a choline head. That choline happens to also be polar, which allows our cells to have that phospholipid bilayer with that hydrophobic area on the inside and then the hydrophilic area on the outside containing its choline head. So triglyceride, 3 fatty acid tails, and then a phos has 2 fatty acid tails plus choline, and we'll say polar head, a polar choline head. That's what a phospholipid has. All right, know the difference between glycogen and starch. Glycogen is actually what animals will store. That's the way we store our sugars. Starch is the way that plants will store their sugars. And remember, plants, when you see cellulose, for example, that's actually a constituent of their cell wall. That's not the way plants are storing their sugars. It's actually the way that plants are using their sugars. So when we store sugars, it's usually we're storing them for energy. So glycogen is the way animals store sugar energy and potential energy, and then starch is the way plants store their sugar potential energy. Difference between dehydration synthesis and hydrolysis. We know that dehydration synthesis is to form bonds by removing a water, and hydrolysis is to break bonds, so hydrolysis by adding water. So lysis means to cut. Hydrolysis is to cut. No difference between saturated fat and unsaturated fat. We know that saturated fats are fully saturated with hydrogen, whereas unsaturated fats have at least 1 double bond, because when you have a double bond, that's 1 less hydrogen that can bind to the fatty acid tail. Be able to recognize the chemical formula of glucose, C6H12O6. Remember, if I'm going too fast, let me know. H12O6, C6H12O6. Be able to recognize the different polysaccharides from their descriptions. Different polysaccharides from their descriptions. Okay, so we're probably talking about cellulose as an example of a polysaccharide. That's going to be used as the plant cell walls and some bacterial cell walls. Glycogen is another polysaccharide. Starch is another polysaccharide. That one's kind of vague, but that's as far as I want you to know. Properties of fat. So if it's unsaturated, it's going to be liquid at room temperature. If it's saturated fat, so most animal fats are saturated, it's going to be solid at room temperature. What does DNA stand for? Deoxyribonucleic acid. Deoxyribonucleic acid. I'm not going to have you spell it on the test, but it's just something to take away for your next class. What are the differences between DNA, RNA? So for the most part, DNA, A, C, T, G, and then RNA has A, C, U, G. Subcomponents of a nucleotide. You got the 5-carbon sugar. You've got the phosphate group, or the phosphate part of the backbone, and then you have the nitrogenous base. 4 general structures of all cells. All cells. So we got ribosome. We've got DNA, or genetic material. We have the juice of the cell, cytoplasm. What else?
Membrane?
Membrane, yes, yes, cell membrane. Awesome, boom. Those are the 4 things, the general structures of all cells. What usually you'll see as a trick on, like, not just my test, but everyone's test, MCAT, PCAT, whatever CAT test you're taking, is going to be, they're going to ask, oh, what about nucleus? But that's a no-go. Nucleus is not a component of all cells, because we know that bacteria are a type of cell. Bacteria have no nuclei. What does fluid mosaic membrane mean? It just means that it's composed of many parts, and it's fluid, so it's constantly in flux, losing, gaining things. What are the properties of phospholipid bilayer, and be able to recognize the hydrophilic polar and the hydrophobic nonpolar regions? So we'll just say that the outside, inside towards cytoplasm equals polar, versus the inside between the outsides. I don't know. Okay, I guess I'll try drawing it. Do you guys get the point? Okay, hold on. I can't just assume you guys get the point. I'll draw it. Here's your phospholipid heads, and just pretend like I'm using two tails on each of these. So in here, in the middle, this is going to be hydrophobic. And then here and here is going to be philic. So phile means a lover of, so philosophy, lover of knowledge. And then phobe means to be afraid of, so this is hydrophobic, hydrophilic. No differences between prokaryotes and eukaryotes cell parts. We know the biggest difference is the lack of nuclei in the prokaryotes. Flagella, pili, and cilia. So flagella, just think of the, sperm will have that long flagella. It's a whip-like structure that actually helps in swimming. And then pili, that's going to be that funnel that bacteria will use to, was it pili? Yeah, that's going to be the funnel that they use to attach to each other. And then cilia, that's going to be like many small flagella, kind of like hairs, and usually that's going to use to move things across the surface of cells. So like your upper respiratory tract has a lot of cilia on the surface of their cells to move mucus up out of your airway. Okay, be able to match cell organelles with their functions. So we kind of went over that. Ribosomes make proteins, mitochondria make energy through oxidative respiration. Your Golgi apparatus is going to repackage stuff, so that's kind of like the post office, and send it out of the cell. Your rough endoplasmic reticulum is going to be involved in, or it provides the perfect housing or perfect environment for protein folding when your ribosomes are synthesizing synthesizing proteins from mRNA. And then your smooth endoplasmic reticulum, that's going to be involved in detox and storage of ions. And what else did we have? Vesicles are just the transport units within the cell. Be able to identify parts of the mitochondria. So the matrix is the inside, and the cristae is going to be those folds. know which type of cells have cell walls and which ones do not. Plant cells and some bacterial cells will have cell walls. Animal cells will not have cell walls. Selective permeability, that just means that if there's a membrane, some things can go in and some things cannot go in. So that's just selective permeability. Know which substances can cross the membrane and which cannot. So small, think smaller, small and non-polar are the easiest to move through the membrane. So ethanol is an example of a non-polar, well actually it's amphipathic. So a lot of drugs are actually non-polar. They can just slip right through the membrane pretty easy. And that's just because if you look at it, your phosphate heads are, not very large compared to your long fatty acid tails. So when it comes to your phospholipid bilayer, there is just simply more volume of the phospholipid bilayer that is nonpolar. And so nonpolar stuff will be able to hang out within that phospholipid bilayer for longer. Be able to distinguish between simple diffusion, osmosis, facilitated diffusion, endocytosis, and exocytosis. As a reminder, if I'm going too fast, let me know. I will slow down. So simple diffusion, that's where things can simply move across the membrane with no requirement of ATP. So that's just, you drop a sugar cube into some water and it dissolves and diffuses. That is simple diffusion. Osmosis, that's where you have a semi-permeable membrane in between or in between separating water from something else. And what happens is the water will have the tendency to move through that semi-permeable membrane from an area of lower solute concentration to an area of higher solute concentration. Remember, solute is the stuff that is not water, that can be dissolved, such as salt, such as sugars. So if you have more sugar on one end of the phospholipid bilayer, water is going to have the tendency to move towards the place with more sugar or more salt. Facilitated diffusion, that's where you actually have special channels in the membranes of cells. We'll just use cells as an example, that allow things that normally wouldn't be able to pass through simple diffusion to pass through that specialized channel. So it's specially shaped to bring in, like sugars, for example. It's got the perfect environment for sugars to pass through it. Endocytosis, that's going to be cell eating, so consuming things, bringing stuff into a cell, and then exocytosis, that is things exiting the cell. And then we already talked about this last time, know how to calculate the total magnification power. We did this on the Kahoot, so you just multiply the two numbers together. So if your ocular lenses, which are going to pretty consistently be 10x, and then you're given any kind of lens, so let's say 100 times, so 10 times 100 is 1,000. If you're given the scanning lens, which is the red lens, that's 4x magnification, so you would multiply 10x times 4x, which would give you 40x magnification. All right, and whenever, this is for the ground, of course, but what power do you use when beginning looking at the specimen? You usually want to use the lowest power, so the scanning lens. All right, let's move on to the next one. Here we go. Okay, no differences between hypertonic, isotonic, hypotonic solutions. So this is all referring to the solution itself. So hypertonic means that you have more solute concentration in the solution than you have within the cell. Isotonic is equal solute concentration outside of the cell as you have inside of the cell. And then hypotonic, that's going to be less solute concentration outside of the cell than you have inside of the cell. Typically, when you have more solute concentration outside of the cell than you have inside of the cell, what happens is that water inside of the cell wants to osmose or move out of the cell, and as a consequence, your cell will shrivel up. Isotonic, your cell should stay the same. Hypotonic, remember, the water wants to move towards where there is more solute, and so if there's less solute outside of the cell, it's going to want to move into the cell, and usually that's where you get the cell bursting. And we call that hemolysis if we're referring to red blood cells, which you'll probably encounter that in your future courses. Be able to compare kinetic energy and potential energy. So kinetic energy is the energy of movement, potential energy is potential energy. So energy that can be used. So ATP is an example of potential energy, or just simply an electron gradient is an example of potential energy, or a ball on top of a hill. That's an example of potential energy. The first and second laws of thermodynamics. So the first one is energy cannot be, energy can't be created or destroyed. And the second law is, the first is energy can't be created or destroyed, only change forms. And the second law is every time you exchange energy, you convert from one energy type to another, you lose some to an unusable form of heat. What is ATP? Adenosine triphosphate. That's what the letters stand for. It's non-energy rich forms. That would be ADP, and it's adenosine diphosphate, or AMP, adenosine monophosphate. Activation energy. So activation energy, that is the energy required to start a reaction. Know what enzymes do and what are enzymes specifically composed of. So if we're talking about, if we're talking biology, most of your enzymes are going to be composed of proteins, mostly proteins. And we know that enzymes are just, we could put biological catalysts. Biological catalysts. What part of an enzyme do substrate bind to? Active site. What determines the enzyme specificity? 3D structure. Because remember, they're proteins. Proteins have three-dimensional structure, and that three-dimensional structure is what determines its specificity. What two primary factors affect enzymes' activity rate? Temperature and pH. What does it mean to denature an enzyme? That's just to mess with its 3D structure. So you give it too high of heat, you start breaking it apart, you start breaking those hydrogen bonds that are holding it together in three-dimensional space. We call that denaturation. Or your pH goes too low or too high, and you're doing the same thing. We call that denaturation. What is a competitive inhibitor versus a non-competitive inhibitor? This will probably help you in the future if you go into the medical field. So competitive inhibitors, I'll write it right underneath. So bind to active site and compete for substrate, or we'll just say compete. It competes for the substrate. Non-competitive inhibitor binds to allosteric site. And remember, when you bind to that site that's not the active site, it changes the three-dimensional shape of the active site so that the substrate, its natural substrate, cannot bind anymore. Be able to write a reaction of respiration and identify which parts are being reduced and oxidized. So we'll just put C6H12O6 plus O2 makes, and actually we'll put 6C6H12O6. We'll get specific here because I don't want to give you guys non-thorough info. Or it's not C6H12O6, it's 6O2. 6O2 to make 6CO2 plus 6H2O plus energy. We'll just say, or ATP. So that's going to be cell respiration. And we know that the things that are oxidized are going to be the things that are that move from a larger structure to smaller structure. So you go from C6H12O6 to 6CO2. That's oxidized. What's reduced are going to be the things that gain electrons. So in this case, you have oxygens, and they gain electrons to make 6H2O. So we'll just put oxidized and then reduced. So this is oxidized, this is reduced. What metabolic processes are used to break down glucose, which require oxygen? I remember this is on the test. So I think I asked something about glycolysis. So that's the first process of glucose breakdown, right? Glycolysis. Used to break down glucose. Okay, so we want to break down glucose down to its base components, which are just carbon in the form of CO2. So glycolysis to pyruvate, oxidation to Krebs cycle, or citric acid. And actually, this is actually what breaks down glucose completely. After that, you're just using those electrons to make ATP. But these are the three processes that will break down glucose. Now, if you're in anaerobic conditions, you go from glycolysis. How do I? Trying to point an arrow going down. Then you could go through fermentation. Right, glycolysis to fermentation, where you take that pyruvate and you ferment it to make ethanol or lactic acid. Define aerobic and anaerobic. I think that's on the test. Aerobic means with oxygen. Anaerobic means without oxygen. A lot of this stuff was on the Kahoot as well. At what step does aerobic cellular respiration begin? That's going to be the pyruvate. That's because in order to send pyruvate into the mitochondria, you need oxygen present, and so that's going to be the first step. Once your pyruvate has entered the matrix of the mitochondria, you've committed to oxidative respiration. What are the two main electron carriers in cellular respiration? NADH plus FADH2. Where does glycolysis occur? Cytoplasm. What is made during glycolysis? Pyruvate. I think that's as far as I want you guys to go. I don't need you to know the specifics. Like I said, it's a little bit more general than the exam that we took online, or the second exam that we took. What two products result from fermentation? So the two products, NAD. And that's important just because that's what we're using to drive forward glycolysis. And then if you're in a mammal, likely lactic acid. What organisms make these products? So anything that can ferment will make, anything that can ferment will make NAD+, will make lactic acid. Yeast will make ethanol. What's required for these products to be made? Low O2, or you could say anaerobic conditions. Okay, it is 1:50. If you guys do want to leave, I won't be offended. I'm just going to keep going until we finish this, though. What happens during pyruvate oxidation? And I'm going to upload this as soon as I'm done too. Or not as soon, because it's going to probably take a while for YouTube to process it. It takes quite a bit. What happens during pyruvate oxidation? Pyruvate oxidation, probably not going to focus fire on that on the test, but know that in pyruvate oxidation, you're taking pyruvate and then you're creating an acetyl group, binding that to coenzyme A, and then you're splicing that coenzyme A into the Krebs cycle. What are the products? So coenzyme A, acetyl coenzyme A, sorry, and then you're also making some CO2. So you're plucking off a carbon in the form of CO2 during pyruvate oxidation. What happens during the citric acid cycle? Oh, that's a lot of stuff, but I'm not going to focus fire on that on the test either. Just know that that's going to be the major period of time, or the major step where you're going to produce the most CO2. So during the citric acid cycle, you're going to make a lot of CO2. You're going to shuffle around atoms until you're going to shuffle around atoms. You're going to strip away electrons. You're going to create some ATP, and then you're going to form the starting product again. What happens specifically to electrons and hydrogen ions in the electron transport chain? So electrons move from electron carrier to electron carrier. That kinetic energy drives forward the pump-to-pump hydrogens into that intermembrane space, creating a hydrogen gradient, a potential energy gradient that ATP synthase eventually uses to create ATP. So what are the results? ATP. How is ATP made by ATP synthase? It uses that hydrogen gradient in the intermembrane space to drive forward the enzyme contained within ATP synthase to make ATP. What are the general yields of ATP of aerobic cellular respiration versus fermentation glycolysis? General yields, so we just know that in aerobic respiration it's going to be higher. In anaerobic, such as fermentation glycolysis, it's going to be lower. Define photosynthesis, so that's going to be the carbon fixation or creating sugars from atmospheric CO2 and light energy. Chlorophyll, that's going to be the pigment, you could say the pigment that plants will use to drive forward photosynthesis. Photons, those are light packets or packets of light energy. And then a proton, I put this in there because they kind of sound the same. A proton is simply a proton. So and you could even think H+, because if you strip away, we know that hydrogen actually doesn't have a neutron. All it has in its nucleus is a proton. So if you strip hydrogen away from its electron, all you're left with is a proton. All you're left with is that subatomic particle. So it's just a proton. Know what products and reactants for photosynthesis. So products are going to be oxygen, so O2, also glucose, right? We're making sugars. And then the reactants are going to be the stuff we're putting into it. So we're going to need light energy. We're going to need water. Remember, that's where the electrons come from in photosynthesis. We're going to need water. We're going to need light energy. And then we're also going to need CO2 from the atmosphere. Be able to write the overall equation for photosynthesis, just the inverse of cell respiration. And instead of ATP, put light energy. What are the sub-steps of the light reaction and the Calvin cycle? Sub-steps of the light reaction. I don't know. I don't think I would break them down into steps. It's kind of like I would just separate the light reaction from the Calvin cycle. So light reaction, that's where you're harvesting light energy to create the intermediate potential energy. So the ATP and NADPH, and then Calvin cycle, that's where you're actually going to fixate or you're going to fix all that carbon that you're pulling in from the atmosphere, and you're going to use that ATP and you're going to use that NADPH that you gathered from the light reaction. Distinguish between the stroma, grana, thylakoid membrane of the chloroplast. So the stroma, that's going to be the juice within the chloroplast. The grana, those are going to be the stack of pancakes. The thylakoid membrane, that's going to be the membrane of every pancake within that stack. And then the chloroplast is going to be the organelle itself. Which color of light do plants? Oh, this is actually on the test right here. This is on the test. And we know that what do plants not absorb as much, therefore reflect? Green. So that would just, it's a freebie on the test. Where in the chloroplast does light reaction occur? Where does Calvin cycle occur? So the light reaction occurs in the thylakoid membrane. Calvin cycle occurs in the stroma. What are the parts of the photosystem? Not going to ask that. What enzyme helps make ATP in photosynthesis? It's going to be the same one that we use in cell respiration, and that is ATP synthase. What is the electron carrier made by the light reaction in photosynthesis? NADPH. This kind of comes in handy because I ask a question where I ask you, okay, what are these things? And then you say, oh, this one looks a little bit unfamiliar, but these two look familiar. But they're actually all familiar because they're all electron carriers. So that's another question. What are the reactants and products of the Calvin cycle? So the Calvin cycle, the Calvin cycle, you're using ATP, NADPH, and then you're essentially making sugar. And then you're not making O2 there. You're not making oxygen there. That's what you do in the light reaction. What are the stomata? What are their functions? Stomata, those are going to be the pores within your plant, within your leaves. So that's how your leaves, your plants will breathe. That's how you get the gas exchange. So that's how you let go of the oxygen and you bring in the CO2. How does the carbon cycle work? So this is just how does the carbon cycle work? You have photosynthesizers that take CO2 from the atmosphere. They make sugars. Then you have heterotrophs that will come and consume the plants. And then what they do is they undergo aerobic respiration. So they're breaking down those sugars and they're taking in oxygen from the environment and they're releasing CO2. And so then that CO2 is brought in by photosynthesizers and so on. Now, alongside industrialization, you know, we have kind of like an overload on one end versus the other end. We have a lot more CO2 production now versus probably less vegetation now. Greenhouse effect, it's the difference between greenhouse effect and global warming. Greenhouse effect is just where you get the buildup of greenhouse gases such as CO2 in the atmosphere. It prevents heat from escaping Earth, and that could lead to global warming. That leads to the heating of the Earth. All right, so that's it.